Assalamualaikum and salam sejahtera. My name is Aidil Eizhar bin Daud. My student ID is 2019 686 748. In this slide, I will brief about what is fetal hemoglobin and adult hemoglobin. First of all, we must know what is hemoglobin. Hemoglobin is the molecule in red blood cells that carries oxygen. One hemoglobin is equal to 4 ions / heme groups, which is a molecule of oxygen. Now the transfer oxygen from mother to fetus. The mother breathes in oxygen and then it's transferred around the body using red blood cell. The red blood cell of adult is slightly different from the fetus. Blood from mother is not able to cross the placenta to get the fetus, but the components in blood such as carbon dioxide and oxygen are able to go through the umbilical cord to the fetus. The movement of the oxygen is dependent on the affinity for hemoglobin. Fetus must have higher affinity to fetal hemoglobin than adult hemoglobin in order to survive. Now is fetal hemoglobin. Fetal hemoglobin is the dominant form of hemoglobin present in the fetus during gestation. HbF is produced by erythroid precursor cells from 10 to 12 weeks of pregnancy. HbF contains 2 alpha and 2 gamma subunit. That genes express gamma chain protein are present in the beta chain locus on chromosome 11. Next is adult hemoglobin. Adult hemoglobin contains 2 alpha and 2 beta subunit. This difference introduces conformational changes to the protein that gives rise to several physiological differences in oxygen delivery that are important in fetal circulation.
Assalamualaikum warahmatullahi wabarakatuh. My name is Fazatul Adawiyah Binti Hamdan and my student ID is 2019241106. Today I would like to explain about comparison dislocation curve between fetal hemoglobin and adult hemoglobin. So, do you know what is oxygen hemoglobin dislocation curve? Oxygen hemoglobin dislocation curve is mean the quantity of oxygen combined with hemoglobin depends on the level of blood PaO2. PaO2 is mean the partial pressure of oxygen. This relationship was expressed as the oxygen hemoglobin dislocation curve is not linear but has a sigmoid curve. Other than that, it has a step slope between PaO2 of 10 to 50 mm Hg and have a flat portion above a PaO2 of 70 mm Hg. The oxygen dislocation curve plot the percent saturation against the partial pressure of oxygen. I will explain about graph oxygen dislocation curve for adult hemoglobin and fetal hemoglobin. From this graph, we can see that fetal hemoglobin curve is more at the left than adult hemoglobin. Fetal hemoglobin and adult hemoglobin has the same percent oxygen saturation, which is 100%. So I will explain more detail about this graph. Now I will explain about adult hemoglobin first. Adult hemoglobin composes 2 alpha and 2 beta chains, while fetal hemoglobin composes 2 alpha and 2 gamma chains. The oxygen dislocation curve for adult hemoglobin is sigmoidal shape due to cooperative binding, and there is a low saturation of hemoglobin when oxygen levels are low, which is hemoglobin releases oxygen in hypoxic tissues. There is a high saturation of hemoglobin when oxygen levels are high, which is hemoglobin binds in oxygen-rich tissue. Now we are going to the fetal hemoglobin. Fetal hemoglobin composes 2 alpha and 2 gamma chains, and the hemoglobin of the fetus has a slightly different molecular composition to adult hemoglobin. It has a higher affinity for oxygen, so dissociation curve is shifted to the left. This is important as it means fetal hemoglobin will load oxygen when adult hemoglobin is unloading. Following birth, fetal hemoglobin is almost completely replaced by adult hemoglobin around 6 months. And lastly, fetal hemoglobin production can be pharmacologically induced in adults to treat diseases such as sickle cell anemia. Thank you, and let's continue to the next speaker.
Assalamualaikum warahmatullahi wabarakatuh and very good day. My name is Mohamad Azharuddin bin Rashid and my ID is 2019203598 and today I'm going to present about the factor that affect the dissociation curve. The first factor is the pH of blood. When more carbon dioxide enter the red blood cell of the capillaries, it is ready to convert into bicarbonate and hydrogen ion and decreasing the pH. The hydrogen ion can bind into electric ion of the hemoglobin and cause a decrease of oxygen affinity. This allows more oxygen to be delivered to the tissue and this phenomenon is called the Bohr effect. A decrease in pH shifts the dissociation curve to the right. Second cause is the temperature of the blood. The cells that have high rate of metabolism produce more thermal energy as products. Thermal energy will transfer to the blood plasma and raise the temperature of the blood. An increase of the temperature will decrease the affinity of hemoglobin to oxygen and will make the hemoglobin more likely to release oxygen to the tissue. An increase of the temperature will cause the dissociation curve to the right. The third cause is carbon dioxide. Metabolically active cells will produce more carbon dioxide as waste byproducts, and some of the carbon dioxide can actually bind directly to the hemoglobin, decrease the affinity for oxygen. Therefore, a higher pressure of carbon dioxide in the blood means that more oxygen will deliver to the tissue. This effect is called as Hamdan effect. An increase of carbon dioxide will shift the curve to the right. The fourth cause is 2,3-biophosphoglycerate or 2,3-BPG. 2,3-BPG is intermediate of glycolysis. When cells have high rate of metabolism, they can produce an excess of 2,3-BPG. Some of 2-BPG can escape into blood plasma and bind to hemoglobin, and it also can lower the affinity of oxygen. 2-BPG can cause the hemoglobin to unload more oxygen to the tissue. An increase of 2-BPG will cause the shift of the dissociation curve to the right. The last factor is carbon monoxide. Carbon monoxide is competitive inhibitor of hemoglobin and it is also 250 times more likely to bind to hemogroup of hemoglobin. By binding to hemoglobin, carbon monoxide causes to increase the affinity of oxygen. Therefore, less oxygen will deliver to the tissue. Carbon monoxide causes the curve to the left, and it also lowers the curve. From the graph, the blue curve is a normal curve, and the red curve will affect with low pH, high temperature, high carbon dioxide, and high 2-BPG. The black curve is affected with high carbon monoxide. Let's continue with the next presenter.
Hi and Assalamualaikum. I Fatin Nabila Binti Mohamad Lateh. In my part, I will explain about the difference between fetus hemoglobin and adult hemoglobin, which are affinity for oxygen, binding with 2,3-bisphosphoglycerates, and stabilizing by 2,3-bisphosphoglycerates. The first difference is fetus hemoglobin has a higher affinity for oxygen rather than adult hemoglobin. This is because fetus hemoglobin is important in transporting oxygen from maternal to fetal circulation. The movement of oxygen from the maternal circulation to the fetal circulation is possible because fetus hemoglobin having a high affinity for oxygen, in contrast to 27 mm Hg for adult hemoglobin, the partial pressure at which fetus hemoglobin is half saturated with oxygen is 90 mm Hg. This value suggests that fetus hemoglobin has a high oxygen affinity, allowing fetus hemoglobin the ability to more readily bind oxygen from the maternal circulation. When fetus hemoglobin has a stronger affinity for oxygen than adult hemoglobin, it can enhance the transport of oxygen within the uterus to the fetus by capturing oxygen from the placental vasculature that has a slightly lower tension of oxygen than in the lungs. The reduction in development approximately 6 months after birth decreases the exchange of oxygen between the blood vessel and other areas of the body, including the brain. Next, fetus hemoglobin binds less tightly with 2,3-bisphosphoglycerides than adult hemoglobin. Fetus hemoglobin also shows a decreased affinity for the metabolic intermediate 2,3-bisphosphoglycerides or 2,3-BPG produced in tissues with high energy uses. In adult hemoglobin, a higher binding affinity to 2,3-BPG induces a right shift, favoring oxygen unloading. The primary structural difference between fetus hemoglobin and adult hemoglobin are located in or near the 2,3-bisphosphoglycerate binding site between 2-gamma interface of fetus hemoglobin and the 2-beta interface of adult hemoglobin. The net result of these structural variations is that 2,3-bisphosphoglycerate binds less strongly to fetus hemoglobin than adult hemoglobin. Next, what is 2,3-bisphosphoglycerate? 2,3-bisphosphoglycerate that binds to hemoglobin is negatively charged. It can decrease hemoglobin's affinity for oxygen. 2,3-bisphosphoglycerate have different amino acids, for example, serine and histidine. Both of them have different ARC group, and ARC group are determine the amino acid function. When fetus hemoglobin binds with 2,3-BPG and serine, it binds loosely, resulting the better binding to oxygen. Next, adult hemoglobin binds with 2,3-BPG and histidine. It binds tightly, causes the bad binding with oxygen. It is because histidine has positive charge, so it will interact well with 2,3-BPG that has negative charge. And the last one is 2,3-bisphosphoglycerate does not stabilize fetus hemoglobin as efficiently as stabilizing adult hemoglobin. Therefore, accounting for the leftward shift of the oxygen saturation curve for fetus hemoglobin compared to adult hemoglobin when measured at the same 2,3-bisphosphoglycerate concentration.
That's all from me, and this part will continue by Fatimah.
Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Binti Salahuddin. My student number is 2019212382. Today I will continue about the difference of fetal hemoglobin and adult hemoglobin. Denaturation of hemoglobin. The denaturation was first conducted by von Kober in 1866. The determination of fetal hemoglobin based upon the resistance of the embryonic pigment to the denaturation by strong alkali. Fetal hemoglobin were resistant to the destructive effects of natrium hydroxide, whereas hemoglobin preparation obtained from adult were rapidly destroyed under the same experimental conditions. Determination of fetal hemoglobin has depended on the either measurement of the denaturation rate under standardized conditions or measurement of the unaltered hemoglobin remaining after denaturation. Next is lifespan. The lifespan of adult hemoglobin for about 100 to 120 days until they fully combine with oxygen or carbon and circulating in blood capillary. Meanwhile, the lifespan of fetal hemoglobin is only 2 weeks. It is because the fetal hemoglobin has a different composition from the adult forms of hemoglobin, which allows it to bind oxygen more strongly. That's all from me. Thank you.
Assalamualaikum and hi, I am Mohamad bin Zaidi. So now, we has reached the climax for this topic. For this part, I will conclude all of the points that has been presented by other presenter. But before that, may I ask you guys, do you guys learn anything from our presentation? If you guys does, I hope this information can help you in your study. As a conclusion, there are a lot of difference between fetus hemoglobin and adult hemoglobin. For instance, their curve shape, their affinity of oxygen, their lifespan, and their binding or stabilizing with 2,3 by phosphoglycerate. As mentioned in this video, fetus hemoglobin has higher affinity of oxygen rather than adult hemoglobin. This is because it helps to enhance oxygen to fetus in womb. Next, fetus hemoglobin has curved shift to left shape. Meanwhile, adult hemoglobin has S shape. Besides, adult hemoglobin has longer lifespan rather than fetus hemoglobin, which is 100 to 120 days. Last but not least, adult hemoglobin has higher affinity to binding with 2,3-bisphosphoglycerate, which provide unloading amount of oxygen. This difference help to determine the characteristic between adult hemoglobin and fetus hemoglobin. I think that's all from us. Once again, I hope you guys understand this topic well and thank you for listening. Bye.